The overall goal of this experiment is to evaluate the effect of the drying air-liquid crystalline, or air-LC interface on orientation of megamolecular biopolymers, such as polysaccharides, microtubules, and DNA as visualized dynamic behaviors in millimeter scale.
This method can help answer key questions in the dispersive structure field, such as self-organization of biopolymers under a drying environment where living organisms have crawled onto land approximately 500 million years ago. The main advantage of this technique is that it allows side views of the sample to be taken during the drying process without relying on the horizontal plane.
To begin this procedure, add 0.5 grams of saccharin to 100 milliliters of water. Cover the container with plastic wrap and stir the solution at approximately 80 degrees Celsius for more than 12 hours. After allowing the saccharin and xanthan gum solutions to cool, centrifuge the saccharin solution to remove impurities. Now, prepare a 0.5 weight % tubulin solution in Britton-Robinson buffer on ice. Mix 50 microliters of the tubulin solution with 5 microliters of GPCPP. Then, incubate the solution at 37 degrees Celsius for 3 hours to obtain a stable microtubule, or MT nucleus. Following this, mix 950 microliters of the tubulin solution and 50 microliters of the GPCPP-containing tubulin solution.